Welcome to Cell and Molecular Biology. Today, we are diving into one of the most fascinating and crucial topics in biology, transcription factors. These are the master regulators of the cell. Without them, our DNA would just be a static instruction manual. By the end of this video, you will understand exactly how these microscopic proteins make the decisions that keep you alive, and how we can even calculate their behavior mathematically. To understand transcription factors, imagine you are standing inside a gigantic, sprawling library. This library represents your cell's nucleus, and the millions of instruction manuals inside are your genes. Now, a muscle cell does not need the instructions for making stomach acid. If the cell tried to read every manual at once, it would be absolute chaos. Transcription factors are the highly specialized librarians of your cells. They know exactly which book to pull off the shelf, when to open it, and when to keep it tightly shut. Now that we have our library analogy, let us look at the formal biological definition. A transcription factor is a specialized protein that binds to specific DNA sequences. Its main job is to control the rate at which genetic information is transcribed from DNA into messenger RNA. To do this, these proteins contain what is called a DNA binding domain. This is a very specific physical shape that perfectly fits into the groove of the DNA double helix at a precise sequence, like a key fitting into a lock. Once these proteins attach to the DNA, how do they actually work? They function as molecular switches. Some transcription factors act as activators. They send out a signal that helps recruit the transcription machinery to the gene, turning it on. Others act as repressors, physically blocking the machinery from attaching, effectively turning the gene off. The primary machine they interact with is an enzyme called RNA polymerase, which is the engine responsible for actually synthesizing the RNA strand. Before we move on, let us see this mechanism with a concrete visual. Here, you are looking at a schematic of the DNA double helix. Notice the specific region labeled as the promoter sequence. The transcription factor, shown binding directly to this sequence, has recognized its specific target. By binding there, it creates a docking station. The arrow shows how it recruits the large RNA polymerase enzyme to the site, allowing transcription to begin. This diagram perfectly captures the activator mechanism we just discussed. Understanding this mechanism leads us to a profound realization about our own bodies. Transcription factors are the fundamental reason why you have different types of tissues. Think about it. Your heart cells and your skin cells contain the exact same DNA. So why do they look and function completely differently? It is because entirely different sets of transcription factors are active in them. They are the master regulators that allow a single set of genetic instructions to create complex, specialized multicellular life. Because these proteins hold so much power, any mistake can be devastating. In the real world, medical researchers study transcription factors intensely. Mutations in the genes that create these factors are a leading cause of many severe diseases, including various forms of cancer. For example, if a transcription factor that normally tells a cell to stop dividing becomes broken or mutated, the cell might multiply out of control and form a tumor. This is why understanding them is critical for modern medicine. Now that we understand what transcription factors are and why they matter, let us look at how they work quantitatively. This formula describes fractional occupancy, which comes from chemical thermodynamics. Y represents the fraction of DNA binding sites currently occupied. TF in brackets is the concentration of the transcription factor in the nucleus. KD is the dissociation constant, which measures how tightly the protein binds. A lower KD means tighter binding. If the concentration increases, Y approaches 1, meaning the gene is fully turned on. Let us apply this formula to a real scenario. Suppose a cell needs to produce a digestive enzyme. We are given a dissociation constant of 10 nanomolar, and the current transcription factor concentration has risen to 40 nanomolar. We want to find the fraction of activated genes. We substitute 40 for the concentration and 10 for the dissociation constant into our formula. Adding the denominator gives us 40 divided by 50. This yields 0.80. Physically, this means 80% of the target genes are currently bound and highly active. Now that we have used the formula to find occupancy, let us tackle a more complex problem, working backward to find the required concentration. Imagine a cell needs to shut down a toxic pathway. We need exactly 95% of the genes turned off, so Y is 0.95. The dissociation constant is 5 nanomolar. We plug these into our standard equation. To solve for the unknown concentration, our first algebraic step is to multiply both sides by the denominator, bringing all terms onto a single flat line. Continuing our derivation, we need to isolate the concentration variable. We distribute the 0.95 into the parentheses, which gives us 4.75 plus 0.95 times the concentration. Remember, the right side is essentially 1.00 times the concentration. To group our variables, we subtract 0.95 times the concentration from both sides. This leaves us with a simple linear equation. 4.75 equals 0.05 times our unknown concentration. We are now at the final step of our derivation. To find the exact concentration, we divide the constant, 4.75, by the coefficient, 0.05. This arithmetic yields exactly 95 nanomolar. This proves that the cell must expend significant energy to produce and maintain a concentration of 95 nanomolar of this repressor protein, just to keep the target gene firmly shut off. Notice how a small increase in desired occupancy requires a massive jump in protein concentration. We have covered a lot of ground today. Let us summarize the most important ideas. First, transcription factors are the ultimate decision makers of the cell, determining which genes are expressed and which remain silent. Second, they function as molecular switches, physically interacting with DNA to either recruit or block RNA polymerase. Finally, we saw mathematically how their binding depends on both concentration and affinity. These remarkable proteins are the true master regulators that make complex multicellular life possible. Great job following along.